Today we are going to be discussing cell division. There are two types of cell division. There's mitosis and there is meiosis. Today we are discussing mitosis, which is the division of body cells. Why do cells need to divide, or why do we need cells to divide? Well, we need them for growth. If your cells didn't divide, you would stay the same size and you never would grow. You are not the same size, more than likely, than what you were last year or 2 years ago. You have probably grown some, and you will continue to grow throughout the years until you reach your full growth potential. Another reason why cells need to divide is to repair. We have this starfish here, and we have someone that has had too much exposure to sun and their cells have been damaged. That division can create new growth for the starfish and new cells for the person that you see on the screen that has gotten sunburn. We also use cell division in reproduction, which we will discuss in our next lesson with meiosis. There are a few key terms that I need you to remember as we go through mitosis and we discuss the phases of mitosis. I need you to remember that all eukaryotic cells, which are cells with a nucleus, store genetic information in chromosomes. Human body cells have 46 chromosomes or 23 identical pairs. Each chromosome is composed of a single tightly coiled DNA molecule. This uncoiled DNA information has proteins in it also, and we call that chromatin. Chromosomes can only be seen when cells are dividing. Until that happens, they are called chromatin, because it's not formed in any way. It's just sort of scattered within the nucleus. But as it goes through the cell division, it condenses in these chromosomes, and we see that for the first time in the cell division process. Chromosomes in dividing cells are duplicated. They're duplicated so that each daughter cell that we end up with at the end of the process has the same information. Their duplicated chromosomes are called chromatids and are held together by the centromere. This middle part is what we call the centromere, and then you have the DNA on each side of the chromosomes. And each individual rod is a chromatid. So you have a chromatid here, the centromere here, but this whole thing together is what we call a chromosome. This is the phases of mitosis, or the stages of the cell cycle. Interphase is the first stage of the cell cycle, and we will talk about that in just a minute. And then you have 4 phases of mitosis. So the first stage of the cell cycle is interphase. Then the second stage of the cell cycle is mitosis. And the third stage of the cell cycle is cytokinesis. And we're going to sort of go over them, but the bulk of what we're looking at today is mitosis. Interphase is the first stage of the cell cycle, and this is very important because most of the life of the cell is spent in interphase. And there's a lot that needs to happen before the division, which happens in the second stage, mitosis. All of these things have to happen. We have to have normal cell functions. We have to have, the cell has to be able to get rid of waste, has to be able to bring nutrients and minerals in. Duplication of the DNA has to take place. So interphase is very important in the stage of the cell cycle because 95% of the cell's life is spent in this stage. Interphase occurs just before mitosis begins. And the DNA is replicated in interphase, which is very important because we need the same information in the cells that we end up with at the end of division that takes place in mitosis. So this DNA replication is very, very important. along with the forming of the organelles that happen during interphase. So interphase is not part of mitosis, but mitosis could not occur if this interphase didn't happen, where the replication of the DNA takes place and the organelles are formed and life functions go on. Basically, it's just living life in interphase, getting ready to prepare to divide in mitosis, which leads us to the heart of our lesson today: what happens during mitosis. The first phase, prophase, chromosomes during this phase, they pair up. And the chromatin, we talked about the chromatin, how it's not really, it's just sort of in the nucleus, but not paired up in any certain way. But in prophase, it starts to pair up and it creates the chromosomes and the chromatids in the nucleus. The nuclear envelope begins to disappear during this and breaks down during prophase. You'll see here that the centrioles and the spindle fibers are on opposite sides of the cell. So it's getting ready to start to what we call line up in metaphase. The chromatids line up at the equator of the cell, or the middle of the cell. The chromatids, those rod structures of the chromosome that we were talking about, are attached to the centromere, that middle part, which attaches to these spindle fibers. So the spindle fibers are attached to the centromere, and these chromatids are lined up in the middle, getting ready to pull apart in anaphase. In anaphase, the centromeres that are attached to those spindle fibers begin to split. The sister chromatids, you have one chromatid pulled this way and one chromatid pulled this way. So that chromosome is pulled apart into individual chromatids, and they move to the opposite pole of the cell. Each chromatid is now a separate chromosome. During telophase, the nuclear membrane and the nucleolus reappear. So now we start to see that nucleus again. And the cell membrane begins to pinch apart. In other words, it elongates and starts to pinch in the middle there, getting ready to separate into two daughter cells. So those are the 4 phases. I'm going to review some of that. Prophase, the first phase of mitosis, the chromatin condenses in the nucleus and begins to visibly form chromosomes, and you can see them under the microscope. And it consists of 2 chromatids held together by the centromere. That's those 2 rod-like structures that are held together by the centromere. Then the second phase we said was mitosis. Metaphase, I'm sorry. Chromosomes line up in the middle. We'll go back to that. They line up in the middle right here. And the centromeres move to opposite sides. Then we have anaphase, where the chromatids separate and being pulled apart by those spindle fibers, and they move to opposite ends. And then we go back to telophase, where the nuclear membrane reappears. We see that nucleus for the first time, and it starts to pinch off in the middle. So we have interphase is the first stage of the cell cycle. That's where it does all of its growing, all of its life functions, getting ready to prepare, and it copies its DNA so that it can divide and both cells can have the same information. Then we go to mitosis, and we have the 4 phases. The last stage, it's not 2 cells yet. It's just before being there, but the last thing that needs to happen in this is the cytokinesis, which is the third stage of the cell cycle. That is when the cell membrane completely divides, forming two daughter cells. So you started out with one and you ended up with two. So this is just an overall look. It's easiest sometimes to think of each step using simple 2 words. So let's look. Interphase. Chromosomes are copied, because that's the most important thing that happens in interphase, is that the chromosomes, or the DNA, the information, is copied. Then we have our second stage of the cell cycle, which is mitosis. Thinking of those 2-word phrases. In prophase, which is the first phase of mitosis, chromosomes condense. So all those chromosomes condense into chromosomes. Then we have chromosomes align in metaphase. So they're pulled together in the middle and they line up in metaphase. So chromosomes align. Then anaphase, 2 words that's easy. Chromosomes separate. The chromatids are being pulled to the opposite sides. And then in telophase, chromosomes relax. The nucleus is formed. They relax and get ready to pinch off. And then in cytokinesis, 2 daughter cells. So daughter cells are formed and the division is complete. This is a chart to help you see in interphase how that chromatin is just sort of in the middle, not formed in any way. But then in prophase, it condenses and forms chromosomes. And you have your centrioles that are on each side with all those spindle fibers. Then in metaphase, our chromosomes are lined up right here. And then our 2 words here for telophase, chromosomes are separated. And then, they're separated. I pointed to the wrong one, I'm sorry. Our anaphase, the chromatids are separated. And then we come over here to our last phase and we see our nucleus now is formed. So telophase, it relaxes and gets ready to be pinched in 2 through cytokinesis and move on to 2 daughter cells, and then it repeats itself and starts all over. Our lesson tomorrow will be on meiosis. We'll see you then.